Hello NEET PG aspirants, welcome back to the PYQ series. The topic for today's video has been requested by several students who find this topic very confusing and difficult to understand and remember. So I tried making a comprehensive video of all the bacterial toxins, and I believe that this video is going to be very helpful for your upcoming PG examination. So let's start with the video. So the topic is bacterial toxin PYQ number 9. So let's start with the MCQ. So the MCQ is, what is the mechanism of action of cholera toxin? So they asked about the mechanism of action of cholera toxin. So option A is inactivation of elongation factor 2, continued activation of adenylate cyclase, inhibition of 60S subunit, and increased guanylate cyclase activity. So what is the answer? So the answer to this question is continued activation of adenylate cyclase. Okay. The first option is that the inactivation of elongation factor 2 is seen by the toxin produced by diphtheria. Okay. And this inhibition of 60S subunit is seen by the toxin produced by, that is the cigar toxin produced by STEC or EHEC. Okay. And then this increased guanylate cyclase activity, this is seen in the toxin produced by the heat stable toxin produced by ETEC. And this activity, that is this continued activation of adenylate cyclase, it is also seen by the toxin produced, the heat labile toxin produced by ETEC. So this cholera toxin and this heat labile toxin, the mechanism of action is similar. Okay, so answer to this question is B. But then how to remember all this? So is there any way, any proper systematic way to approach this type of topic so that you will be able to remember it and recall it? So let's see. So what I have done, so this toxin may be divided on the basis of the structure. There is structure like protein, polysaccharide, or maybe on the basis of the bacteria producing like diphtheria toxin, pertussis toxin, like this. And or it may be classified or categorized on the basis of the mechanism of action. So what I will do, I will discuss this topic on the basis of mechanism of action. So I have categorized the toxins on the basis of their mechanism of action. Okay, and the toxins are adenylate cyclase toxin, ADP ribosylating toxin, deamidating toxins, glucosylating toxins, metalloprotease toxins, and RNA glycosidase toxins. You may find it a little confusing or little difficult to understand that, okay, how I am going to remember these toxins now on the basis of their mechanism of action. So don't worry, once you will complete this video completely, you will be able to remember and understand all the toxins with their mechanism of action, with the receptor, everything very easily. So just stay with the video and don't panic. Okay, so I'll start with this adenylate cyclase toxin. So in adenylate cyclase toxin, there are included three toxins. The first one is adenylate cyclase toxin, second is the edema factor, and third is exo-Y. So I'll start with the first, that is adenylate cyclase toxin, which is produced by Bordetella species. Okay, so the structure of this adenylate cyclase is composed of one A subunit and one B subunit, and it does not require any receptor, so it will directly enter into the cell. So direct entry into cell, no receptor is required for this. Okay, and this adenylate cyclase, which is released by this bacteria Bordetella species, will remain inactivated and it will require activation, and this activation is mediated by calmodulin. So this activation of adenylate cyclase occurs in presence of calmodulin, and thus once it is activated, it results in cyclic AMP production, as we know, in the host cells, and then cyclic AMP mediated effects depending upon the cell type. Okay, so this is the first toxin of this adenylate cyclase toxin, easy to remember. Now the second, which is edema factor or EF, uh, and it is also mentioned a plus protective antigen. What does it mean? So this Bacillus anthracis produces a toxin known as anthrax toxin. Okay, and anthrax toxin has got three components. So the first one is EF, second is PA, and third is LF. EF is edema factor, PA is protective antigen, and LF is lethal factor. So this protective antigen will either act with EF or with LF or both. So that what does it mean? That individually alone these three components will have no toxic effect. Alone these have no toxic effect alone. Okay. However, if this EF, that is edema factor, if it is associated with that PA, that is protective antigen factor, it will become toxic. Or if this LF is associated with PA, it will become toxic. So this EF, which is associated with protective antigen, is an adenylate cyclase type of toxin. So the structure is composed of this A and this B. This B is PA and this A is EF. Okay, so these are the two subunits of this toxin, and the receptor where it acts is anthrax toxin receptor, that is ATR. And the mechanism is similar to that of Bordetella. So remember, this Bacillus and Bordetella will have similar mechanism of action, the toxin produced by them. Now this XOY, which is an exoenzyme Y produced by Pseudomonas aeruginosa, it has got only one A subunit and there is no B subunit, and the receptor for it is mediated through type 3 secretion. So it does not need any receptor. However, it enters inside the cell through a type 3 secretion system. So the secretion system or secretory system is of total 7 types, 1 to 7 types of secretion system. Okay. So out of these 7 types, the Pseudomonas aeruginosa utilizes type 3 secretion for entry of this exoenzyme Y into the cell. And this activity is similar to that of adenylate cyclase toxin, that it will increase the cyclic AMP production. However, it does not need any kind of activation. That means it is calmodulin independent. So only difference between the adenylate cyclase and edema factor from the XOY is that XOY does not require calmodulin for its activation. Okay, so these are the adenylate cyclase toxin. Okay. Okay, so now let's see the next type of toxin, that is ADP ribosylating toxins. So this contain the maximum number of toxin produced by the bacteria. So it has got these 4 and then these 3 type of bacterial toxins. So they have got total 7 type of ADP ribosylating toxins. So the first one which is asked in the question is cholera toxin, which is produced by Vibrio cholera. And the structure of this cholera toxin is composed of 1 A subunit and 5 B subunit. A and B, I'll tell you what does that mean. The A is basically the active subunit and responsible for the activity of the toxin, and the B subunit is binding subunit, which is not only responsible for the binding of the toxin to the cell, but it also facilitated the entry of the active subunit inside the host cell. Okay, so it has got two activity, first binding and then second is facilitation of entry of A subunit into the cell. It is similar for the previous also which I have discussed, that ADP adenylate cyclase toxins. Okay, so the receptor for this cholera toxin is GM1 ganglioside receptor. So the ganglioside which is responsible for the entry of cholera toxin is GM1 ganglioside. Okay, and how it will act? It will do ADP ribosylation. So what it will ribosylate? It will ribosylate the GS alpha subunit of G protein. Okay, so GS alpha subunit, that is alpha subunit of G protein. What is G protein? G protein is basically guanine nucleotide binding protein, which actually helps in transmitting signal from outer or exterior to the cell to the interior of the cell. So it is basically a signal transmitting protein. Okay, so once this ADP ribosylation of this G protein, or specifically the GS alpha protein in case of cholera toxin, it will activate the host cell adenylate cyclase. And once this adenylate cyclase, what will happen? Similar to that of Bordetella, what we have seen, that it will increase the cyclic AMP level. In this case, the cell which is involved is the intestinal cell, so it will lead to cyclic AMP mediated fluid secretion. Okay, so ultimately it will lead to activation of adenylate cyclase and increases cyclic AMP level. Okay, and then cyclic AMP mediated fluid secretion. However, the initial step is the ADP ribosylation of GS alpha protein. Okay, this heat labile toxin or LT, which is produced by E. coli, as I told you in the question itself, that has got similar action that of CT, that is cholera toxin, and the structure is also similar, that A and 5B subunit, and also the receptor is also similar, that is GM1 ganglioside. However, it will also bind weakly to the surface glycoprotein, and the mechanism of action is similar to cholera toxin. So what is the difference between these two is the potency of the toxin. This cholera toxin is almost 100 times more potent than heat labile toxin produced by E. coli. So this is the only difference between these two. Okay. Now the third one is diphtheria toxin. It is also an ADP ribosylating toxin produced by Corynebacterium diphtheria, and it has got only one A and one B subunit. And the receptor is heparin binding epidermal growth factor like factor, that is HB, EGF and CD9 receptors. Okay. And what it will do? It will do ADP ribosylation of EF2. What is EF2? Elongation factor 2, which is actually responsible for translocation of ribosomal messenger RNA. Okay. So once it will get ribosylate, that it becomes inactivated, so it will ultimately inhibits the protein synthesis. Okay. And thus causes cell death. So the mechanism of action is ADP ribosylation of EF2, elongation factor 2. Now this exotoxin A, which is produced by Pseudomonas aeruginosa, exo Y, we have seen in adenylate cyclotoxin. So Pseudomonas aeruginosa produces another toxin known as exotoxin A, which has got structure similar to that of Corynebacterium diphtheria toxin, but the receptor is different. That is alpha 2 macroglobulin receptor or low density lipoprotein related protein receptor related protein, that is LRP. Okay, so this alpha 2 macroglobulin or low density lipoprotein receptor related protein. Okay, and the action is similar to that of diphtheria, so that it will also do ADP ribosylation of elongation factor 2 and inhibit the protein synthesis and causes cell death. So these two have got similar mechanism of action. This diphtheria toxin and, okay, let me change the color, diphtheria toxin and exotoxin, and these two have got similar mechanism of action, that is cholera toxin and heat labile toxin. Okay. Now that the other three toxin which have got similar mechanism of action, that is ADP ribosylation, are Bordetella pertussis toxin and the toxin produced by Clostridium botulinum, that is C2 and C3 toxins. Okay. So this pertussis toxin, the structure is similar to that of cholera toxin and heat labile toxin, that is it has got one A and 5 B subunit, but the receptor is glycoprotein. Okay, and what it will do? It will do the ADP ribosylation of the heterotrimeric G protein. Here also it will do ADP ribosylation of G protein similar to that of cholera toxin, in which the ribosylation is of GS alpha protein. Okay, and as I told you, this G protein is responsible for transmitting signal from exterior to interior of the cell. So it will lead to blockage of signal transduction mediated by those proteins. Okay. So this is the mechanism of action of the pertussis toxin, that is ADP ribosylation of several heterotrimeric G proteins. C2 toxin produced by Clostridium botulinum and C3 ADP ribosyl transferase toxin produced by Clostridium botulinum both have got similar structure, that is composed of one A and one B subunit. However, the receptor in for C2 toxin is glycoprotein, while the C3 toxin enters via pinocytosis. Okay, and the C2 toxin will leads to ADP ribosylation of G-actin protein, which is a globular actin protein which is responsible for cellular organization, and thus it result in inhibition of polymerization of actin and causes cell rounding. So cell rounding effect is because of C2 toxin. And this C3 toxin leads to ADP ribosylation of Rho protein. What is Rho proteins? Rho proteins are basically proteins belongs to RAS superfamily. Okay, and what is this RAS superfamily? This RAS superfamily are the family of proteins which is responsible for organelle development. So this ADP ribosylation of Rho protein causes their inactivation and then causes cytopathic changes in the target cell. So as it is required for organelle development, cellular development, so once it is inactivated, it will leads to cytopathic changes in the target cell. So these are the mechanism of ADP ribosylating toxins. Okay. Now, the another type of toxin, that is glucosylating toxin, and only one toxin produced by Clostridium difficile will act in this way, and these are the toxins A and B. So Clostridium difficile produces two toxins, toxin A and toxin B. These are two separate toxins, and the structure is for both of them is one A and one B. Okay, and for toxin A the receptor is glycoprotein. However, for the toxin B the receptor is unknown. So, and the mechanism is, it will lead to, instead of ADP ribosylation of rho protein, it leads to monoglucosylation of rho protein, and thus inactivation leads to cytopathic changes in target cells, similar to that of C3 toxin produced by Clostridium botulinum. So Clostridium difficile toxins and Clostridium botulinum C3 toxin will act on rho protein. The difference is only the mechanism. It will lead, C3 will lead ADP ribosylation, and this will lead monoglucosylation. However, the effect is same, that is cytopathic effect on the target cell. Okay. Now there are certain metalloprotease toxins, and the two metalloprotease toxins are basically neurotoxins which acts on the neurons, and these are botulinum toxin and tetanus neurotoxin. This botulinum toxin, tetanus neurotoxin, the structure is same, that is 1A and 1B. So if you see that most of the toxins have got 1A and 1B subunit. The only toxins which have got more than 1B subunit is cholera toxin, heat labile toxin, and pertussis toxin, which has got 1A and 5B subunit. Okay, and the receptor for this botulinum toxin is GT1B and GD1A gangliosides, and probably also synaptotagmin. Okay, and for this tetanus neurotoxin it is GD1B. So GD1A is the receptor for Clostridium botulinum, and GD1B is receptor for tetanus neurotoxin. Okay, so how it will act? So it will lead to cleavage of snare protein. What is snare protein? Snare protein or snare complex is basically a complex which is responsible for fusion of neurotransmitters containing vesicles. So if you see, just I'll try to make a diagram. So if suppose if this is the synaptic end and this is the presynaptic end, okay, terminal, and suppose if here the neurotransmitter containing vesicles are present. Okay, neurotransmitter containing vesicle. So this neurotransmitter containing vesicle will fuse with the presynaptic membrane, and then it will release the neurotransmitter which is responsible for synaptic transmission. Okay, so this fusion of this neurotransmitter containing vesicle with the presynaptic membrane is mediated by this snare protein. So this fusion is mediated by snare protein. So once if there is a cleavage of this snare protein, this fusion will not occur, and thus it will block the release of neurotransmitter. So in case of botulinum toxin, it will block the release of acetylcholine and thus leads to flaccid paralysis. However, in case of tetanus neurotoxin, this cleavage of this snare protein complex result in blocking of neurotransmitter containing glycine and GABA, not the ACH as in case of botulinum. So what it will lead to? It will lead to spastic paralysis because this neurotransmitter will act on inhibitory terminal. So it will not act on inhibitory terminal. So it will lead to spastic paralysis. So this is the difference. So what are the toxin which inhibits the protein synthesis? The first one is cigatoxin and cigalactoxin, which indirectly prevent the EF1, that elongation factor 1, which is responsible for binding of amino acid tRNA with the ribosome, but this occurs by inhibition of 60S ribosomal subunit. The diphtheria toxin and the exotoxin A produced by Pseudomonas, this will also inhibit the protein synthesis but by ADP ribosylation of EF2. So EF2 is with these two, diphtheria toxin and exotoxin A. EF1 is with cigatoxin and cigalatoxin, but then EF1 is not directly inactivated in the case of cigatoxin. However, in case of diphtheria toxin, EF2 is directly inactivated by ADP ribosylation. So this is the difference. Okay. So now the third metalloprotease toxin, that is lethal factor. This toxin is, as I told you in the first one, that is adenylyl cyclase toxin, that anthrax toxin has got three components, EF, PA and LF. So this LF is a metalloprotease toxin along with that PA, okay, not alone. Remember it. Alone these three components has no activity. So it will need this protective antigen. So this LF along with PA is a metalloprotease toxin, and the structure is again is two separate protein. One is the A, which is the lethal factor, and second is the B, which is the protective antigen. Okay, and the receptor is similar to that of that inhaled cyclase toxin, that is edema factor is anthrax toxin receptor. And what it will do, it will leads to cleavage of MAPKKs. What is that MAPKKs? MAPKKs is basically mitogen activated protein kinase kinases. Okay. So this mitogen activated protein kinases, when it will cleave, it will lead to cell death. But how it is leading to cell death, that pathway is still unclear. Okay, so only remember that this lethal factor acts by cleaving MAPKKs. Okay, so these are the metalloprotease toxins. Okay, so now coming to the RNA glycosidase toxins. So this includes two toxins. One is SIGA toxin and another is SIGA-like toxin. SIGA toxin is produced by Shigella dysentery, and its structure is similar to that of cholera toxin or heat labile toxin, that is composed of one A subunit and 5 B subunit. And the receptor is globotriaosylceramide, in short GB3. Okay, and how it will act? It will lead to single site depurination of basically adenine residue, okay, of 28S ribosomal RNA within the 60S ribosomal subunit. So within this ribosomal subunit, this 28S ribosomal RNA, there will be a single site depurination of adenine residue, and thus what it will leads to, it will actually inactivate or prevent the EF1, which is responsible for the binding of amino acyl tRNA to ribosome. So this binding of amino acyl tRNA with ribosome will not occur, and ultimately it will inhibit the protein synthesis and thus causes cell death. Similar action is seen by the Shigella or Shiga-like toxin produced by Escherichia coli, EHEC, and this Shiga-like toxin resemble the toxin produced by Shigella dysentery type 1. Okay, and its structure is again similar to that of Shiga toxin and cholera toxin and heat labile toxin, and the receptor is similar to that of Shiga toxin, that is GB3, and also the mechanism. Okay, so the next toxin is de-amidating toxin. These toxins are produced by E. coli. The toxin produced by E. coli is CNF1, that is cytotoxic necrotizing factor 1 and 2, and by body tailor pertussis it is dermonecrotic toxin. And how it will act? It will leads to de-amidation of raw proteins and thus inhibits its GTPase activity. But the clinical significance of these toxins is still not known. Okay. Heat stable toxin, as I told you in the discussion of the question itself, heat stable toxin which is produced by just EHEC, and there are two types of heat stable toxin, STA or ST1 and STB or ST2. So this STA, it basically causes secretory diarrhea. Okay, and how it will lead to secretory diarrhea? It will bind to GCC receptor, and what is that GCC receptor? Is guanyl cyclase C receptor. Okay, so by binding to this GCC receptor, it will activate obviously guanyl cyclase enzyme and thus increases cyclic GMP. This time cyclic GMP increases and leads to outpouring of the fluid and thus leads to secretory diarrhea. This STA or ST1 is also produced by Yersinia enterocolitica, non-oven Vibrio cholera and Saccharobacter fructi, but again their role in diarrheal illness is unclear. This STB or ST2, it is a pore forming toxin, so it will form pore in the cell membrane. Okay, but the mechanism is not clear that how it is actually leading to cell death, but it is obviously not through cyclic AMP or cyclic GMP. Okay, So these are the heat stable toxin produced by EHEC. Now, apart from there are certain toxins which does not have any enzymatic activity. So these are pyrogenic exotoxin. They do not possess any enzymatic activities. Okay. And what it will lead to, it will stimulate the T cell proliferation by interaction with class 2 MSC along with antigen presenting cell, class 2 MSC on antigen presenting cell. Okay. And it will really leads to release of massive amount of inflammatory cytokines such as IL-1 and TNF. Okay. And thus these class of exotoxin or toxin are also known as super antigens. So these are super antigens. Okay. And what it includes? It includes this TSST. What is that TSST? This is toxic shock syndrome toxin, which is responsible for toxic shock syndrome. This is produced by Staphylococcus aureus. Another toxin produced by Staphylococcus is staphylococcal enterotoxin, which is responsible for food poisoning. Then another toxin which is produced by Staphylococcus is exfoliative toxin, which is responsible for staphylococcal scalded skin syndrome. Okay. And then there are certain pyrogenic toxins which is produced by Streptococcus species like pyogenes or group B, group C, group F and group G, which are also super antigen. And this pyrogenic exotoxin produced by Streptococcus pyogenes is responsible for toxic shock syndrome. So this is asked in a question that which of the following is super antigen. So the option was not any of these three toxins, but it was the pyrogenic exotoxins produced by Streptococcus pyogenes. Okay. So these are the toxins which do not have any enzymatic activities. Apart from this STB or ST2, there are certain more pore forming toxins, and if you see, the most of the pore forming toxins are lysine toxin, like the listeariolysine produced by Listeria monocytogenes, streptolysine O produced by Streptococcus pyogenes, pneumolysine produced by Streptococcus pneumoniae, and this Panton Valentine leucocidin. Apart from this TSS, exfoliative toxin and the enterotoxin, Staphylococcus aureus also produces a toxin known as Panton Valentine leucocidin, which is a pore forming toxin. And there are certain neurotoxin which causes diarrhea, and this neurotoxin is ingested as pre-formed toxin. That means these toxins are produced in the food before its consumption, and these include the staphylococcal enterotoxin, bacillus cereus emetic toxins and the botulinum toxin. And where it will act, this will act on central nervous system and stimulate vagus, and this was discussed in the PYQ of food poisoning, so that's why I'm not discussing it here anymore. Okay. So these are the various type of toxins produced by bacteria. So answer the question. All of the following staphylococcus infection is toxin mediated except. So as I told you, the pyrogenic exotoxin, the TSST, causes toxic shock syndrome. Staphylococcal scalded syndrome is caused by exfoliative toxin. Food poisoning is caused by staphylococcal enterotoxin. All of these are toxin mediated except the septicemia, which is basically because of presence of the bacteria in the blood. So this is not toxin mediated. So answer is septicemia. So the next MCQ, a 25-year-old man who recently suffered a puncture wound in his leg developed generalized muscular spasm and curved back. Which of the following mechanism of action of the toxin or poison is responsible for this type of posture? So there is a history of punctured wound. Okay. And then the development of generalized muscular spasm and curved back. That means the spasm or the paralysis is spastic paralysis. Okay. And this history of punctured wound indicates the contamination of wound with the spores of a very famous organism causing spastic paralysis, as I discussed earlier in this video, is Clostridium tetani, and what it will leads to production of tetanus toxin. And this tetanus toxin, how it will act, if you remember, it will leads to cleavage of snare protein complex and thus inhibit the release of neurotransmitter GABA and glycine. One thing I have not told you during that the inhibition is presynaptic. What I told you that this snare protein leads to fusion of neurotransmitter containing vesicle with presynaptic membrane. So these toxin, both the botulinum toxins and this tetanus toxin, will act on presynaptic terminal because of this reason only. So this cleavage of snare protein and presynaptic inhibition of glycine and GABA release leads to spastic paralysis. So this is the correct answer. So what is the first one? This cleavage of snare protein blocking ACH release. This is the mechanism of action of botulinum toxin. Okay, botulinum toxin. And this post-synaptic inhibition of glycine and GABA release, this is because of strychnine poisoning. Strychnine poisoning and tetanus toxin infection, the only difference is the site of action, that it will act on post-synaptic terminal rather than on presynaptic terminal. So it will leads to spastic paralysis, but the site of action is post-synaptic. And then this D option has cleavage of snare protein and presynaptic inhibition of ACH receptor. This is again botulinum. Okay, so the correct answer is C. So I hope I was able to explain this topic and try to make comprehensive but easier way to understand the various bacterial toxin. But this is the complete. So you will don't have to worry about that any toxin is left. So I have included all the toxins. Just go through this video 2, 3, 4 times. Then only you will be able to understand and then make a note from this video. And definitely I tell you that you will never miss any question. You will never mistake any question related to bacterial toxin. Okay, so all the best, take care.